So coenzyme requirement.
So it's A, right?
That increases with increase in protein intake, plays a key role in glycogenolysis, and loss of memory. So it is a vitamin B6. Okay. So what vitamin B6 means?
Thiamine?
No,
It's
Pyridoxine.
yes, vitamin B6 means
it's B.
there is PLP. Vitamin B6 means PLP. So B is the correct answer. Okay. Now next question. A person having delayed wound healing might have deficiency of which of the following cofactor?
Zinc.
Yes, zinc. So these are not in part of lecture that I taught, but you can see this is zinc. So this you can take as a very important information and question as well. So a person cannot tolerate pH drop below 6.8 because of
I think it's B, but I could be wrong.
so it's enzyme activity is cannot be very high below, because within certain pH range only enzyme is active. And then you can see enzyme activity is low. That's good. Thank you. So B is the enzyme activity is very low. Okay, and that is why.
Okay.
So you can see interference with other enzyme denaturation of the body. So this also happens. So why does this happen? Because of ultimately denaturation of the enzyme actually the sentencement it is a bit wrong in the sense enzyme denaturation will happen. Okay, so enzyme denaturation, and then because of that activity of enzyme is low. Okay, so you can B and D both are correct.
Can you explain why the other one was zinc?
Tell me.
Can you
Other one is zinc.
like why was this one?
So zinc is part of DNA polymerase, an enzyme that causes polymerization of DNA and even RNA polymerase. These enzymes require zinc, and even some proteases require zinc. So zinc, it has been also seen that if zinc is deficient, then what happens is healing is delayed. Okay, you got that?
Yeah,
Yes.
yeah.
Okay, so in last class I also told you one thing, Q10. Do you remember what is the concept of Q10?
Yeah, it's something to do with like
Yes.
synthesizing fatty acids, and
It's not. It's about the enzyme activity.
I just
So
did.
what does
The
rule
temperature
of
coefficient?
Q10 say? Yes, tell me.
Is Q10 the temperature coefficient?
Yes, it is related to temperature. If you increase temperature by 10 degrees Celsius, the reaction
The
rate
rate
will
of reaction
double.
doubles.
Yes, this is the rule of Q10. Remember this rule about temperature. Got that? Rule of Q10 and optimum temperature and pressure. Optimum temperature and pressure. Temperature, not pressure, pH. Optimum temperature and optimum pH for enzyme. So enzyme requires an optimum temperature and optimum pH for its normal function, for its peak function. At optimum temperature and pH, enzyme will has peak activity. So this concept, very simple concept, one concept optimum, and then this concept. These two concepts are very important. And then why reason This gives you reason why pH, decrease in pH or increase in pH causes reduced activity. So these are important concept about temperature and pH. Concept of optimum temperature pH, rule of Q10, and why. Why pH drop or pH rise decrease the activity. Why temperature rise, temperature decrease the activity of enzyme. Got that?
Does coenzyme Q10, I'm sorry, is it so it basically it just converts ATP?
No, it's no, Q10 is different. Quotient, Q for quotient, with quotient of 10 degrees Celsius, reaction rate double.
Okay.
That does it mean. It is not coenzyme Q.
Okay.
Okay, it is different from coenzyme Q. Q10 means quotient with quotient of 10, reaction rate doubles.
Okay.
Got that?
Yep.
Okay. So these are
Doctor,
important.
there are some people waiting to let them in.
See, this today I'm allowing. So few students are there who actually is not getting kicked out, but they are late. So from next time I will not allow. Already 12 minutes late. So this suffers. This actually a great problem for everybody. So from next class I will not allow people more than 5 minutes. Okay, for up to 5 minutes I will allow. After 5 minutes I will take attendance. Then those people which are out, I will not allow them. So few people they are always late. Okay. So
Doctor, answer to the last question.
Yes.
What was the answer to the last question? The one with pH?
So this question is a practice question. So you can say B and D. You can both have the answer. So enzyme activity becomes low. Why? Why does enzyme activity becomes low? Because of denaturation of enzyme. So I am giving both answer together.
Okay, thank you.
Okay, got that? Okay.
Yes, doctor.
So these are practice question. So I am doing my way so that you will have more information than usual.
Are these questions expected in the quiz on Monday?
Yes, these questions, see, this one, I will convert this one question into case-based scenario. Okay, I will put some stream over there and the same type of question I can ask in one line. Okay.
Okay. In this question, the slide you just showed about TPP.
No, there will not be two answers in exam.
Yeah, I know that. But the answer which you showed TPP about,
Here,
this
is
one.
PLP. This is pyridoxal phosphate, not TPP. This is B6, vitamin B6.
Yeah, B6 is thiamine, right? Thiamine?
No, this is thiamine. This is B1.
Okay.
This is TPP. This is B6. This is whenever you increase the intake of protein, you have to increase the intake of vitamin B6. Vitamin B6 is related to metabolism of protein, amino acid. You have to remember one thing. Actually, I will elaborate in MD2. And upcoming session also, I will talk about vitamin. Repeatedly in metabolism, I will talk about vitamins as well. Okay?
Okay. Thank you so much.
Okay. You're welcome. So you can remember this as a concept right now. It will help you. I will reinforce it. If you memorize right now, I'll reinforce it repeatedly. Let's talk.
Can you go over C and D quickly too? FMN and NAD, like the other vitamins.
So please, you go through those slides. It is there.
Okay. B3.
Okay. This FMN is vitamin B2, and this is vitamin B3. Okay. And you can go through the slides I have given you. There are 2-3 tables, and you have to memorize those all tables. Okay. So those tables are very important. Actually, I will be repeating. If you have memory, it will be repeat for you. But if you don't have memory of those tables, so you will have you will see every time coming the same thing, and you will be confused. Okay. Those are important things. Okay. These vitamins are very, very, very important things. I will be teaching you this about these vitamins from the beginning. Right from the first day, I'm teaching you vitamin. I'll keep teaching vitamin till the end. So that important this vitamin is. Okay. Okay. So I will move to the next topic. So enzyme, today we'll talk about enzyme kinetics and regulations. First we will talk about enzyme kinetics, then we will talk about enzyme regulation. So see, so we have did properties of enzyme. There are S3 and I3. So S means speed, selectivity, and specificity. And you know what I have already described. What is this selectivity? It selects a particular substrate. And then we already talked in detail about specificity. Types of enzymes we did about the type of enzyme. Different enzyme have different specificity. That means different enzyme has different types of reaction. They catalyze specific type of reaction. We have done this specificity in detail. Now we will be talking about this speed. So kinetics means speed. So we are talking about the speed. Today we will be talking about the speed. How speed of enzyme changes with when you increase the substrate concentration. So we will be studying about that today. And then regulation. This among these 3R, 1R we have all 2R we have already studied. And then last R we will study about regulation. So in that sense, we are just elaborating the properties of enzyme. So you can use this picture as the whole to describe whole thing as mnemonics or visual mnemonics as a summary also. So you can use, so you have to reaction condition. In case of reaction condition, you have to memorize, you have to conceptualize this graphs into your mind. For we will talk about this speed kinetics also. There also we will describe about the graph. So you'll have to put those graphs into your mind. Now for comparison, we will use this kinetics for comparison also. So there also graphs come. So this is related. These two are related. Kinetics, this comparison of this enzyme kinetics are related. Enzyme inhibition. It is. We will talk about enzyme inhibition. These are related. Now specificity we did in very detail about specificity. Here is the vitamins, minerals. So these are very important part of vitamins and are very important part of the enzyme that are present at the active site or reaction site. Reaction site also called substrate. Reaction site is not the actual name you will find using. You will find active site or substrate site. I have only named it as reaction site because reaction happens there. And in order to give this mnemonics so that you will remember S3 and R3. So let's talk about today's topic, enzyme kinetics. Kinetics means speed. So enzyme kinetics deals with the speed of enzymatic reaction. And then enzyme increases the rate of reaction. Why? By decreasing the activation energy. Actually, this statement is not precisely true. Okay, this statement is not precisely true. This statement says enzyme increases the rate of reaction through decreasing activation energy. No enzyme can decrease activation energy. The precise statement will be enzyme increases rate of reaction by choosing a pathway having low activation energy. So this will be the most appropriate statement. Enzyme will increase the rate of reaction by choosing a different pathway that have low activation energy. Enzyme nothing has that capacity to decrease some energy. So enzyme cannot decrease activation energy. Rather, enzyme chooses a different pathway that have low activation energy. So you will find such a statement. Why I have written this statement here? Because you will find such a statement in many books. But in Lipin cuts, you will find this statement. Lipin cut, you will find this statement. Enzyme chooses a different pathway that have low activation energy. So this is the most appropriate explanation why enzyme increases the rate of reaction. I will elaborate this statement in more detail with picture, upcoming picture. I have those pictures. So how does enzyme do this? So let's elaborate this. You can see the enzyme. So this is a previous pathway. So you are doing something. You are going this side. You are doing some work after by climbing the whole mountain and reaching there. But what does enzyme do? Enzyme does not do this. Enzyme creates a tunnel in the whole of this pathway in the mountain, and through this tunnel, what will happen? Reaction, the activity will be very easy. So this is what enzyme does. Enzyme chooses a different pathway than the original pathway, which require less energy. Okay, which require less energy to do job. So this is the function of enzyme. Enzyme chooses a tunnel pathway. It does not choose this pathway. So that is the significance of enzyme. This is how enzyme increases the rate of reaction by this lowering the activation energy. This is how it lowers the activation energy. Did you understand this explanation? How enzyme increases the rate of reaction?
Doctor,
Yes.
so enzyme depend on the sites as at the same time depend on the lower energy for activation, right? We can say like this if we have multiple choice question.
So, can you repeat?
Yeah, we can like select multiple choice question if they ask that enzymes choose the best site to interact
Yes.
with lower energy for activation.
Yes, that is the most appropriate choice. That would be the most appropriate choice.
Okay.
And if you don't find that choice, and if the choice have like enzyme lowers activation energy, then that can be second appropriate choice. Okay, that is why I have written both sentences. You can see this statement here, and next statement you will find here. Okay, so this is the most appropriate statement, because I have explained this. You know this. I have already explained this from this picture. Now let's move forward. So you can see here, basically enzyme follow this pathway. This is the curve between this enzyme energy requirement and then enzyme. So it is the pathway of reactant and product. So you can see this is a pathway, original pathway, and this is the pathway of enzyme. So enzyme does this. So enzyme follows a pathway having low activation energy. This is the pathway having low activation energy. So this is how enzyme increases the rate of reaction. Increases the rate of reaction. So this is the mechanism how enzyme increases the rate of reaction. So this is the kinetics. Kinetics means speed. So how does enzyme increases the speed? Enzyme increases the speed by choosing a pathway having low activation energy. Now let's see. So
it's kind of like people then. It's kind of lazy, right? It chooses the most efficient path to.
So see what happens. You can say you can call us people lazy also, because like we are reducing too much of time by talking with mobile phone with each other.
Yeah.
So in the previous time, if you had to talk, you had to go at the place the people live.
Right.
So in that sense, we were wasting too much of time.
Okay. Yeah. Got you.